Hi class, this is your instructor, Scholar Huff. This is Chapter 36, Transport in Plants. Terrestrial plants face two major challenges: maintaining water and nutrient balance, and providing sufficient structural support for upright growth. The vascular system transports water, minerals, and organic molecules throughout the plant, sometimes over great distance. The giant sequoia, Sequoiadendron giganteum, pictured here, grows to an average height of 50 to 85 meters tall. That's 165 to 280 feet tall. Unlike in woody plants, where growth to great heights can be supported by secondary growth of vascular tissues, in herbaceous plants, it is water balance that keeps plants upright. Here, you will learn how water, minerals, and organic molecules move between the roots and shoots of short and tall plants. How does water get from the roots to the top of a 10-story high tree? Plants lack muscle tissue and cannot pump fluid through a circulatory system. Nevertheless, water and dissolved solutes move through the cell wall spaces between the protoplasts of cells, through plasmodesmata, across plasma membranes, and through the interconnected conducting elements extending throughout a plant using a variety of mechanisms. Water first enters the roots, then moves to the xylem, the innermost vascular tissue of plants. Water rises through the xylem because of a combination of factors, and most of that water exits through stomata in leaves. The movement through the body of plants from roots to stems is unidirectional. The greatest distances traveled by water molecules and dissolved minerals are in the xylem. Water moves across cell membranes of root cells into the xylem in the stem. Once water enters the xylem of a redwood, for example, it can move upwards as much as 100 meters. Most of the force that moves the water is pulling, caused by transpiration. And that is the evaporation of water from stomata, for those that may not have remembered that. This pulling occurs because water molecules stick to other, called cohesion, and I'll say again, it's because of water molecules sticking to each other. That's cohesion, and to the walls of the tracheate or xylem vessel, which is called adhesion, due to hydrogen bonding. This result is an unusually stable column of liquid that can extend for great distances. Long-distance transport also occurs in the phloem. Unlike xylem transport, phloem transport is bidirectional. If a single plant cell is placed into a hypotonic solution, the concentration of solutes inside the cell is greater than that of the external solution, and water moves into the cell by the process of osmosis. The rate of osmosis across a membrane, however, is limited. For a long time, scientists were puzzled when water was found to move more rapidly than predicted by osmosis alone. We now know that the rate of water movement into or out of cells is enhanced by membrane channels called aquaporins. These transport channels occur in both plants and animals, and you were taught this in Principles of Biology 1. In plants, they exist in vacuoles and plasma membranes and allow for bulk transport across membranes. Aquaporins speed up water movement, but do not change its direction. They are important in maintaining water balance within a cell and in moving water into xylem. Water potential and pressure gradients form a foundation for understanding local and long-distance transport in plants. Plant biologists explain the forces that act on water within a plant in terms of potentials. Potentials are a way of representing free energy, the potential to do work. Water potential is abbreviated by the capital Greek letter psi, with the subscript W, is used to predict the direction in which water will move. The key to remember is that water will move from a cell or solution with a higher water potential to a cell or solution with a lower water potential. Water potential is measured in units of pressure called megapascals. If you fully turn on your kitchen or bathroom faucet, the water pressure should be between 0.2 and 0.3 megapascals, which is 30 to 45 psi. The total water potential of a plant cell is the sum of its pressure potential and its solute potential. It represents the total potential energy of the water in the cell. And here's the equation I was about to refer to. So total water potential equals the sum of its pressure potential and its solute potential. And that's the total potential energy for the water in the cell. So if you look closely here, and this is in your textbook from figure 36.4, C, the total water potential of a plant cell, as you see it here, is calculated just like this. Now on to water absorption. Most of the water absorbed by the plant comes through the region of the root with root hairs. Root hairs are those extensions of root epidermal cells located just behind the tips of growing roots. Surface area for the absorption of water and minerals is further increased in many species of plants by associations with mycorrhizal fungi. These fungi extend the absorptive net far beyond that of root hairs and are particularly helpful in the uptake of phosphorus in the soil. Once absorbed through the root hairs, water and minerals must move across cell layers until they reach the vascular tissues. Water and dissolved ions then enter the xylem and move unidirectionally throughout the plant. Water and minerals can follow three pathways to the xylem of the root. The apoplast route includes movement through the cell walls and the space between cells. Transport through the apoplast avoids membrane transport. The symplast route is the continuum of cytoplasm between cells connected by plasmodesmata. Once molecules are inside, they can move between cells through plasmodesmata without crossing a plasma membrane. The membrane transport route involves membrane transport between cells and also across the membranes of vacuoles within cells. Of the three routes, the transmembrane route allows cells to carefully regulate what solutes enter and exit the cell. These routes are not exclusive, and molecules can change pathways anytime until reaching the endodermis of the root. Eventually, on their journey toward the root xylem, molecules reach the endodermis. Any further passage through the cell walls is blocked by the Casparian strips. All cells in the cylinder of the endodermis have connecting walls embedded with the waterproof material suberin. Molecules must pass through the plasma membranes and protoplasts of the endodermal cells to reach the xylem. The endodermis, with its unique structure, along with the cortex and epidermis, controls water and nutrient flow to the xylem to regulate water potential and helps limit leakage of water out of the root. Mineral ion concentration in the soil water is usually much lower than it is in the plant. Minerals are often taken up by root cells via active transport across the endodermis to increase the solute concentration in the steel. The plasma membranes of the endodermis cells contain a variety of protein transport channels through which proton pumps transport specific ions against even larger concentration gradients. Once inside the vascular steel, the ions, which are plant nutrients, are transported via the xylem throughout the plant. The aqueous solution that passes through membranes of endodermal cells enters the cell's vascular tissues and moves into the tracheids and vessel members of the xylem. As ions are actively pumped into the root or moved via facilitated diffusion, their presence decreases the water potential. Water then moves into the plant via osmosis, causing an increase in turgor pressure. Root pressure, which often occurs at night, is caused by the continued accumulation of ions in the roots at times when transpiration from the leaves is very low or absent. This accumulation in an increasingly high ion concentration, which in turn causes more water to enter the root hair cells by osmosis. Ion transport further decreases the solute potential of the roots. The result is the movement of water into the plant and up the xylem columns, even though transpiration is not pulling the water upward. Under certain circumstances, root pressure is so strong that the water will ooze out of a cut plant stem for hours or even days. When root pressure is very high, it can force water up to the leaves, where it can be lost in a liquid form through a process known as guttation. Guttation cannot move water up great heights or at rapid speeds. It does not take place through stomata, but instead through special groups of cells called hydathodes located near the ends of small veins that function only in this process. When you see drops of water called dew on plants' leaves in the morning, this is largely due to condensation of water vapor in the atmosphere. However, some water may be the result of guttation. Root pressure alone, however, is insufficient to explain xylem transport. Transpiration provides the main force for moving water and ion solutes from roots to leaves. Tracheids and vessel elements are essential for the bulk transport of minerals. Ultimately, the minerals that are actively transported into the roots are removed and relocated through the xylem to other metabolically active parts of the plant. Phosphorus, potassium, and nitrogen, and sometimes iron may be abundant in the xylem during certain seasons. In many plants, this pattern of ionic concentration helps conserve these essential nutrients, which may move from mature deciduous parts such as leaves and twigs to areas of active growth, namely meristem regions. Keep in mind that minerals that are relocated via xylem must move in the generally upward flow through the xylem. Not all minerals can re-enter xylem conduit once they leave. Calcium, an essential nutrient, cannot be transported elsewhere once it has been deposited in a particular plant part, but other nutrients can be transported in the phloem. More than 90% of the water taken in by the roots of a plant is ultimately lost to the atmosphere. Water moves from the tips of veins into mesophyll cells, and from the surface of these cells it evaporates into pockets of air in the leaf. These intercellular spaces are in contact with the air outside of the leaf via stomata. Water is essential for plant metabolism, but it is continuously being lost to the atmosphere. At the same time, photosynthesis requires a supply of CO2 entering the chlororenchyma cells from the atmosphere. Plants therefore face two conflicting requirements: the need to minimize water loss to the atmosphere and to take in carbon dioxide. Structural features such as stomata and the cuticle have evolved in response to these requirements. The rate of transpiration depends on weather conditions, including humidity and time of day. As stated elsewhere in this section, transpiration from the leaves decreases at night when stomata are closed and the vapor pressure gradient between the leaf and atmosphere is less. During the day, sunlight increases the temperature of the leaf, while transpiration cools the leaf through evaporation. On a short-term basis, closing the stomata can control water loss. This occurs in many plants when they are subject to water stress. But the stomata must be open at least part of the time so that CO2 can enter. As CO2 enters the intercellular spaces, it dissolves in water before entering the plant's cells, where it is used in photosynthesis. The gas dissolves mainly in water on the walls of the intercellular spaces below the stomata. The continuous stream of water that reaches the leaves from the roots keeps these walls moist. The two sausage-shaped guard cells on each side of a stoma stand out from the other epidermal cells, not only because of their shape, but because they are the only epidermal cells containing chloroplasts. Their distinctive wall construction, which is thicker on the inside and thinner elsewhere, result in them bending when they become turgid. Turgor in guard cells result from active uptake of potassium and chloride and malate. As solute concentration increases, water potential decreases in the guard cells and the water enters osmotically. As a result, these cells accumulate water and become turgid, opening the stomata. Protons are pumped out of a guard cell by action of light-activated photon pump. This creates a charge difference across the membrane, which causes ions to move into the guard cell. The guard cells of many plant species regularly become turgid in the morning when photosynthesis occurs and lose turgor in the evening, regardless of the availability of water. During the course of a day, sucrose accumulates in the photosynthetic guard cells. The active pumping of sucrose out of the guard cells in the evening may lead to a loss of turgor and close the guard cell. Transpiration rates increase with temperature and wind velocity because water molecules evaporate more quickly. As humidity increases, the water potential difference between the leaf and the atmosphere decreases. But even at 95% relative humidity in the atmosphere, the vapor pressure gradient can still sustain full transpiration. On a catastrophic level, when the whole plant wilts because of insufficient water availability, the guard cells may lose turgor, and as a result, the stomata may close. Fluctuations in transpiration rate are tempered by opening or closing stomata. Experimental evidence indicates that several pathways regulate stomata opening and closing. Abscisic acid, ABA, plays a primary role in allowing potassium to pass rapidly out of guard cells, causing these stomata to close in response to drought. CO2 concentration, light, and temperature also affect stomata opening. When CO2 concentrations are high, the guard cells of many plant species close the stomata. Additional CO2 is not needed at such times, and water is conserved when the guard cells are closed, although the exact mechanism is still being investigated. Blue light regulates stomata opening. This helps increase turgor to open the stomata when sunlight increases the evaporative cooling demands. Some plants store CO2 at night in a modified form to be used in photosynthesis during daylight hours. This crassulacean acid metabolism, or CAM, photosynthesis, has wide distribution and has been found in plants of 40 different families, including desert plants such as cacti. In this process, stomata open and CO2 is taken up at night and fixed into organic acids. These compounds are decarboxylated during the day when stomata are closed, providing a source of CO2 for fixation by the usual Calvin cycle. This allows CAM plants to conserve water in dry environments. Because plants cannot simply move to another location when water availability or salinity changes, adaptations allow them to respond to such environmental fluctuations. Many mechanisms for controlling the rate of water loss have evolved in plants. Regulating the opening and closing of stomata provides an immediate response to changes in water availability. Morphological adaptations provide longer-term solutions to drought periods. For example, for some plants, dormancy occurs during dry times of the year. Another mechanism involves limiting transpiration by loss of leaves. Deciduous plants are common in areas that periodically experience drought. Thick, hard leaves with relatively few stomata, and frequently with stomata only on the lower side of the leaf, lose water far more slowly than large pliable leaves with abundant stomata. Leaves covered with masses of woolly-looking trichomes reflect more sunlight, thereby reducing the heat load on the leaf and demand for transpiration for evaporative cooling. Plants in arid or semi-arid habitats often have their stomata in crypts or pits in the leaf surface. With these depressions, the water surface tensions are altered, reducing the water loss. The search is on for genes responsible for drought tolerance. Plants can also receive too much water, in which case they ultimately drown. Flooding rapidly depletes available oxygen in the soil and interferes with the transport of minerals and carbohydrates in the roots, often resulting in abnormal growth. Hormone levels change in flooded plants. Ethylene, a hormone associated with suppression of root elongation, increases, while gibberellins and cytokines enhance growth of new roots, usually decrease. The hormonal changes contribute to the abnormal growth patterns. Oxygen deprivation is among the most significant problems because it leads to decreased cellular respiration. Standing water has much less oxygen than moving water. Generally, standing water flooding is especially harmful to a plant. Flooding that occurs when a plant is dormant is much less harmful than flooding when it's actively growing. Physical changes that occur in roots as a result of oxygen deprivation may halt the flow of water through the plant. Paradoxically, even though roots of the plant may be standing in water, its leaves may be drying out. Plants can respond to flooded conditions by forming larger lenticels and adventitious roots that reach above flood level for gas exchange. While some plants survive occasional flooding, others have adapted to living in fresh water. One of the most frequent adaptations among plants to growing in water is the formation of the arenchyma, which is loose parenchymal tissue with large air spaces in it. Arenchyma is very prominent in water lilies and other aquatic plants. Oxygen may be transported from other parts of the plant above water to those below by passages in the arenchyma or the arenchyma. This supply of oxygen allows cellular respiration to take place even in the submerged portions of the plant. Some plants normally form arenchyma, whereas others, subject to periodic flooding, can form it when necessary. In corn, increased ethylene due to flooding induces arenchyma formation. And this shows what arenchyma look like. They're in the middle with those large air spaces from the loose parenchyma tissue. The algal ancestors of plants adapted to a freshwater environment from a saltwater environment before the move onto land. This adaptation involved a major change in controlling salt balance. Plants such as mangroves that grow in areas normally flooded with salt water must not only provide a supply of oxygen to the submerged parts, but also control their salt balance. The salt must be excluded, actively secreted, or diluted as it enters. The black mangrove has long, spongy, air-filled roots that emerge above the mud. These roots, called ematophores, have large lenticels on their above-water portions through which oxygen enters. It is then transported to the submerged roots. In addition, the succulent leaves of some of the mangrove species contain large quantities of water, which dilute the salt that reaches them. Many plants that grow in such conditions also either secrete large quantities of salt or block salt uptake at the root level. Most carbohydrates manufactured in leaves and other green parts are distributed through the phloem to the rest of the plant. This process is known as translocation. Provides suitable carbohydrate building blocks for the roots and other actively growing regions of the plant. Carbohydrates concentrated in storage organs such as tubers, often in the form of starch, are also converted into transportable molecules such as sucrose and moved to the phloem. Here, it will be discussed ways by which the carbohydrate and nutrient fluid term sap is moved through the plant body. The movement of sugars and other substances can be followed in phloem using radioactive labels. Radioactive carbon from carbon dioxide, that's 14, can be incorporated into glucose as a result of photosynthesis. Glucose molecules are used to make the disaccharide sucrose, which is transported in the phloem. Such studies have shown that sucrose moves both up and down in the phloem. The most widely accepted model of carbohydrate transport through phloem is the pressure flow hypothesis. Dissolved carbohydrates are loaded into the phloem at the source, and the flow from the source to the sink where they are unloaded. Carbohydrate sources include photosynthetic tissues, such as the cortex of roots can be either sources or sinks. Sinks also occur at the growing tips of roots and stems and in developing fruits. Sources and sinks can change across the life of a plant. As needs change, the direction of phloem flow can shift. In a process known as phloem loading, carbohydrates, and mostly sucrose, enter the sieve tubes in the smallest veins at the source. Some sucrose travels from the mesophyll cells to the companion and sieve cells via symplasts. Much of the sucrose arrives at the sieve cell through apoplastic transport and is moved across the membrane using secondary transport employing a sucrose hydrogen ion symporter. Companion cells and parenchyma cells adjacent to the sieve tubes provide ATP energy to drive this transport. Unlike vessels and tracheids, sieve cells must be alive to participate in the active transport. Bulk flow occurs in sieve tubes without additional energy requirements because of the difference between the water potential in the sieve tubes and that in the nearby xylem cells. Water flows into the sieve tubes by osmosis. Turgor pressure in the sieve tubes thus increases, and this pressure drives the fluid through the plant's system of sieve tubes. At the sink, sucrose and hormones are actively removed from the sieve tubes and water follows by osmosis. The turgor pressure at the sink drops, causing a mass flow from the turgor pressure at the source to the weaker pressure at the sink. Most of the water at the sink then diffuses back into the xylem, where it may either be recirculated or lost through transpiration. Transport of sucrose and other carbohydrates within sieve tubes does not require energy, but the pressure needed to drive the movement is created through energy-dependent loading and unloading of these substances from the sieve tubes. This has been your instructor, Scholar Huff.